We are continuing with the topic of biotechnology. Yesterday, we clearly understood how gene therapy or genetic modification or genetic engineering occurs, how the process will be carried out, what are the different steps involved, what are the different enzymes that we require at various stage, that everything we learnt in detail. Today, we are progressing ahead with few more. These things I am not going to explain now in detail, but these are certain other aspects which you will remember as the topic goes by. Right. We know about DNA. DNA is there in nucleus and nucleolus. Again, that gets transferred to offspring, that means next generation. 23 chromosomes from the mother, 23 from the father, 23 pairs of chromosomes will be there in the offspring. That means child. Up to this we are very clear. There is another kind of DNA is also available in human body, that is called mitochondrial DNA. Don't get confused. Mitochondrial DNA, unlike nuclear DNA, is only a single chromosome will be there in mitochondrial DNA. Whereas nuclear DNA, you have two chromosomes, you have 23 pairs of chromosomes will be there. And here it is only each mitochondria will get a single chromosome, will have a single chromosome. This particular chromosome will not have any role in genetic information or the characters of the offspring child. The inheritance happens only with male and female chromosomes. Mitochondrial DNA will not have any role in genetic information. Clear? But why mitochondrial DNA should be present? Yes, mitochondrial DNA helps in producing proteins. Those are required for functioning of the metabolism of an individual. Metabolism, that means the bodily functions. The proteins required for these bodily functions will be produced by mitochondrial DNA. All characters, all traits of the child will be by mother and father, but the metabolic aspects, the proteins produced by the DNA of mitochondria, metabolic aspects, metabolic proteins will be produced. Simply, this is the difference between nuclear DNA and mitochondrial DNA. But what is a mitochondria? In the cell, you will have a nucleus, a nucleolus, like that. It is a cell organelle. That means a part of a cell, a small part in the cell which will have a certain function is mitochondria. This mitochondria is the storehouse of energy for human cells. Whatever energy that is produced, that will be utilized for the functions, bodily functions, will be stored in the form of ATP, adenine triphosphate, ATP. ATP gets converted into energy. Energy will be utilized by the bodily organs of the human being. So you can call mitochondria is the storehouse of energy of every cell. It is a cell organelle. I think you have understood what is a mitochondrial DNA and what is a chromosomal DNA. But you will ask me, why you are telling us about mitochondrial DNA? Yes, mitochondrial DNA, what happens? The sperm comes from the father, the ovum will be in the mother, sperm sperm enters the ovum, fertilization happens. That means then onwards, it will be there in the mother's womb. Father will not do anything with that. Further developmental process of the embryo has to happen in the mother's womb. Mother's womb, that means no, mother's ovum will have some more cells or some mitochondrial cells which will produce mitochondrial DNA. That means the proteins which are required for future bodily functions will be produced by the mother mitochondria. Though characters of father and mother get expressed in the offspring, the metabolic activities of the embryo or future child will be from the mother only. Mother mitochondria contributes the metabolic mechanism. Proteins required for metabolism will be from the mother's ovum. That means mother's mitochondria will only do the functions of producing proteins required for bodily metabolism in the embryo or subsequently in the offspring. Father only contributes to the sperm, which is the genetic material which will not have mitochondria. Mitochondria comes from the mother. That means protein for the metabolism of the baby comes from the mother. Understood? So, what if the mother develops some diseases related to metabolism? That means, apart from the characters, expressions, mother's mitochondria is diseased, and it will produce some deleterious effects. It will not function the metabolic activities very effectively. Such kind of a mitochondria from the mother is going to the embryo, and subsequently the individual develops mitochondria related diseases because mother's mitochondria is having such kind of diseases, and mother's mitochondria is contributing to the child. In this scenario, what happens? Automatically, if you are not controlling or if you are allowing the mother's mitochondria produce proteins required for the metabolism of the embryo, automatically the diseases of the mother will get carried. If you are stopping mitochondria, the proteins will not be produced at all. That is again a big problem. Here you need to do the diseased mitochondria will have to be replaced by the healthy mitochondria. For this, you need to know what is a mitochondrial DNA. Okay. The significance of mitochondrial DNA you have understood. There is a concept called three-parent baby. The three-parent baby is nothing but mother is one part, mother's chromosomes is one part, father's chromosomes is another part. That means no, those are the parents, two parents. When diseased mitochondria of the mother is replaced by the healthy mitochondria of a donor who is not the biological mother, but whose healthy mitochondria only you have taken to substitute the diseased mitochondria of the mother, then the baby thus produced will become a three-parent baby. Here, mother is one parent, father is another parent. The donor who have given mitochondria, healthy mitochondria to the embryo will become the third parent. none of the characters of the donor will come to the offspring. Only the proteins which are required for effective metabolism of the embryo and subsequent development of the child, only there the role of the donor comes. However, this particular technique is called a three-parent baby. Understood? Here, in the three-parent baby, again I tell you, only the mitochondria of a donor is taken, none of the other characters are taken. Right? In significant cases where it will be affected, once the embryo is formed, from the embryo, you will introduce the healthy mitochondria of a donor into the embryo, and then you are again inserting, once you are introducing a healthy mitochondria into the embryo, and then thus produced embryo with the healthy mitochondria is again placed into the uterus of the mother. Then remaining process of developing the baby into different stages, that all happens in the womb of the mother. Thus generated baby will be called a three-parent baby. This is a concept which, no, this is simply a concept which you will remember. Then this all happens in vitro. That means in vitro means in the lab. In vivo means within the body. In vitro, you are taking out the embryo, and in the lab you are inserting healthy mitochondria, and then you are again placing the embryo into the womb of the mother. That's why the process is called an in vitro procedure. Then we learn about stem cells. What are these stem cells? You nowadays science is talking too much about stem cells. What are stem cells? How they are different from normal cells? Yes, the stem cells, you will have normally in a body, there will be 220 kinds of cells. Each cell is responsible for multiplication and then forming an organ, forming a tissue. Tissue again forms an organ. Organ again forms a body. Likewise, many organs now will form the ultimate body. What are the tissues? Your bone cell is a kind of a tissue. It produces the only bone cell. It will not produce any other cell. Your muscle cell. The muscle cell, the mother muscle cell, start producing only muscle cells. It will not produce a bone cell. Likewise, a nerve cell. A nerve cell, mother nerve cell, start producing, multiplying into many nerve cells. It will not produce any other cell. Likewise, each cell is very specific in nature, very specific in function. It will not produce any other cell but for the set of cells now which it is producing. Likewise, a tissue will be produced by that muscle cell, muscle cells. Then muscle, wherever no muscle is required in the arm, leg, wherever muscle is required now, that particular muscle will be formed. In this muscle, there will be many kinds. There are parenchyma, collenchyma, many kinds of tissues, soft tissues, hard tissues, many are there. Likewise, in the body, a total of 220 types of tissues are present. Muscle, bone, blood, nerve, immune cell, white blood cell, and sex cell, epithelial cell. These are all various types of cells. Cells, those form tissues which in turn become organs. This is the general cells. The cells of two different kinds in the human body. These cells, as far their function is concerned, there are two types of cells. One is differentiated cells Second is undifferentiated cells. The very meaning of the word itself, no, you will know differentiated cell means it it turns, it will know it produce same type of cell, many cells, ultimately it becomes a complex tissue becomes an organ. These are differentiated cells. In science terminology, differentiation means producing more number of similar kind of thing is differentiation. Undifferentiated means here undifferentiated cell is not meant for one single type of cell. This undifferentiated cell can form any kind of a spell. I told you 220 types of cells. These undifferentiated cells can form according to the requirement of the body. It can produce any kind of a mother cell. From that mother cell, that particular kind of a tissue will be formed. And in other words, an undifferentiated cell can produce, is capable of producing a bone cell, capable of producing a nerve cell, capable of producing a blood cell. Likewise, all kinds of 220 kinds of cells can be produced by an undifferentiated cell. But a differentiated cell, if it is meant for producing a muscle tissue, it produces muscles only. Likewise, it is very specific. Where undifferentiated cells would be present, where they need to be. Undifferentiated cells will be in the embryo before the development starts, before the organ starts. It's only when in embryo stage the cells will be undifferentiated, most of the cells, because in the embryo you can't say which is a leg, you can't say which is a, in the beginning stages no organ is formed, no organ, at least initiation did not happen. Then the cells of such embryo are undifferentiated cells. That means they can produce any kind of a cell depending upon the requirement of the embryo. As the embryo is developing into various stages, the undifferentiated cells will contribute to production of a specific cells, differentiated cells. From there, the tissue organ happens. Another area, I told you embryo is the one area where undifferentiated cells can be located. There is another area where undifferentiated cells will be present. This is bone marrow. So, two areas. One is embryo, second is a bone marrow. These undifferentiated cells will be present. These undifferentiated cell can form any kind of a cell. There is no restriction that it has to produce only this kind of a cell. It can produce all kinds of cells of the body when it is required, where and when, what kind of a quantity is required, that everything no body demands according to the requirement. This undifferentiated cell acts as a particular cell of that area. So these undifferentiated cells, which have a capacity of producing any kind of a cell, are called stem cells. These are very primitive cells. Why it is primitive means from the beginning, from the embryo stage onwards, you will have these cells. In the advanced stage, you will find in the bone marrow, when embryo will develop into a particular structure, a definite with limbs and all kinds of development happens in an embryo. These different undifferentiated cells, you can no more find them, but you will find in the bone marrow. That's the only area for a grown adult. You will find stem cells in the bone marrow. But if you want to deal with an embryo, stem cells will be present in the embryo, right? Then embryonic cell, stem cells is available, and in adult cell, so some somatic cells are there. Somatic means no only physiological activity. somatic cell will be there. Somatic cell is not a stem cell, but with some kind of a treatment, you can convert a somatic cell into, you will not be able to convert a somatic cell into an embryonic cell, but you can convert a somatic cell by a kind of an external treatment. You can convert a somatic cell into a stem cell, and you can use it as a stem cell when it is required. This is all advanced technology. When you are applying some advanced technology, then only a somatic cell can be transformed into a stem cell. Otherwise, the naturally occurring somatic cells are present in the embryo as well as in bone marrow. The nature of stem cell producing into any kind of a cell, any kind of a cell complex is called, the nature is called totipotency. What is totipotency? Totipotency is the nature of the stem cell to transform itself into any kind of a cell, any kind of a cell. That is totipotency. That means even in the embryo, if you are damaging some cells, automatically it will regenerate, form new cells very quickly so that the damaged part can be covered. This happens very quickly, very fastly in the embryo, but as it appears as no, in the suppose bone marrow, if you are disturbing taking out a sample of bone marrow also gets recuperated. Whatever amount you have taken, again the bone marrow generates new bone marrow cells and it compensates for it, but it takes time. It will not happen as quickly as it is happening in a embryo. It is simple. The children, you see how fast they are, and the elders know the fastness comes down. Cloning of entire organism can also happen with a single cell. That means no, it has got a totipotency. That means the stem cell can be modified into any kind of a cell. If you want to develop an organ in the lab, suppose a body, a tissue which no the required for a limb, want to generate a cell, stem cell from the embryo can be extracted, and it can be it can be placed in the lab conditions. It can be made to produce the tissue required to fill the limb, arm, or whatever limb no you wanted to do that. Similar kind of a tissue can be produced. So this is about stem cell. Stem cell introduction is that stem cell is that you can any embryo, defective embryo is there, new embryo cells can be again no, in the process of gene therapy it can be taken, it can be kept here, and then no, the new embryo without any defect no, it can be formed. There is another cell called IPS. It is induced pluripotent stem cell. It's a technique, in fact. It's not a naturally occurring cell. It is called induced pluripotent stem cell, IPS cell. It will be just like a stem cell which you are now taking out either from the embryo or the umbilical cord or from the bone marrow. It is just like the function of such kind of a induced pluripotent stem cell will be just like any other naturally occurring stem cell. It will be used in therapeutic activities and particularly cloning to treat a disease, degenerated diseases. Degenerative diseases, there will be some of the bodily related degenerative diseases are there. Muscle related diseases, nerve related diseases. What happens? This induced pluripotent stem cell, that means it is just like a stem cell. You can take out, you can induce from the somatic cell also. This pluripotent stem cell can be induced by external technique, and you can induce such kind of a pluripotent stem cell into the embryo. Then the replacement for the diseased, degenerated, the kind of diseases happens. defective embryo cell you are taking out, you are injecting an induced pluripotent stem cell. So it just acts as a natural stem cell, healthy stem cell, and it covers up for the disease. Disease will not get transmitted to the offspring. Wherever in the normal process also, after a adult happens to get a particular disease related to a stem cell based disease, that means no any cell complexes of these 220 kinds, any damage noticing, then you will induce and then repair that particular cell damage by introducing externally a pluripotent stem cell and then repairing the problem there. It can, you can do that. It need not be done in the embryo stage. It can be done in the adult stage also. Cells that can be transformed to stem cell to replace the diseased cell. What is the significance of stem cell therapy? You see baldness, blindness, deafness, bone marrow related diseases, and then Alzheimer's disease, osteoporosis, Parkinson, and slowing of the such kind of diseases. All these diseases you can cure all these diseases with the help of the stem cell therapy or by induced pluripotent stem cell therapy. Stem cell therapy is to replace, even in the adult, any dysfunction of the stem cell related activity. You can rectify it. Parkinson, you cannot rectify the disease, but you can slow the disease. Now, it is vigorously expanding. Probably you can slow the pace of it by pushing it to a few more years. The forgetfulness or no such kind of traits, you can effectively control them. You cannot effectively control them totally, but you can slow the disease by, you can push it by a few more years. Another technique I am telling, this is growing human organs in animal body. Humans are different, animals are different. Growing human organs in animal body. This is not for any reason. This is just to see whether we can grow any organ, any human organ, whether there is a possibility. You want to see whether there is a possibility. Through experiments, can we produce a human organ? Can we grow or cultivate a human organ? It's not by manures, not by agriculture, but these therapeutic measures through this stem cell therapy or stem cell therapy mechanisms, can we do that? What is the need for that? Suppose a man is continuing with a diseased kidney, a diseased liver, a diseased kidney, let's take. The kidney need to be given by a donor. In order to make him healthy, somebody has to donate the kidney, and there should be some many matches. DNA fingerprinting will be done, and then no blood samples matching will be done. Many kinds of tests will be taken before somebody accepts a donor. Now, instead of asking for donation, can we grow the similar kind of a kidney with what kind of a kidney he is suffering? Can we grow in a period of time similar kind of kidney somewhere and then excise it, cut it, and then can we, in the place of donation, can we use thus grown kidney into this man? These kind of an experiments now for the future, vigorously people are doing this kind of an experiment. Particularly Japan is very advancing with these kind of techniques. What they have done is that till now, they have grown a mouse kidney in a rat using the stem cell. What they have done? They have taken out the embryo of the rat successfully. The gene that is responsible for eliminated. In that place, a mouse stem cell is taken and then inserted in this place. Ultimately, when they saw the stem cell was induced to produce a kidney, ultimately this embryo is again placed into the womb of the uterus of the rat. Then after some time, in the rat, a kidney of a mouse was produced. That means the experiment is successful. They want to do similar kind of an experiments with relation to human organs. The experiments are going on. Probably in another 5 to 10 years, we don't know, we may hear that we can grow human organs somewhere, and then now you can keep them a store as a standby. When you find a defect in yourself, probably for you where it is grown, you can take out, and then no, that will be a standby procedure. All your kidneys, liver, such kind of things will be grown at some place. Your number is 22. 22 individuals' organs are cultivated there. They have kept them there. By chance, the kidney of 22 happened to be diseased. It is no more functioning. They will take that out. And you have grown your standby organ somewhere. You will take it out like a vegetable. You will bring it here, attach it here like a donation. Ultimately, here this man functions in a much better way. There are 10-15 years ago, there were in a fiction, English fiction, you have found out these things. There is very a wealthy group of individuals who are millionaires, billionaires were there. Of course, they were numbered. What they have done is that some scientists have come to their rescue in a secluded place. They have started cultivating pigs, and all these matching organs of these identified wealthy individuals, there they have given a number to each pig. In each pig, they started growing the replication of the organs related to one particular individuals. That is very secured and wonderful place where they are growing these things. Whenever this particular individual is falling sick or one of his organs is not functioning, the idea is to excise an organ from that numbered pig and bring it here. 15 years ago, this was in the fiction. Now it is coming to the reality. Happening, experiments are happening like that. So this is a kind of a technique, and in the rat, mouse kidney was formed. That means there is a guarantee that in the future the human organs can also be grown. Then I am going to gene editing. Gene editing is nothing but a kind of gene therapy which I have discussed yesterday. Gene editing is in a sequence, some defective gene is, you have identified a defective gene. You are using CRISPR or Cas9 enzyme, and then you are cutting that particular defective gene, taking out, and then you are replacing with a healthy gene. That is one method. Second is called gene silencing. The defective gene will be taken out, and if it's not related to any particular expression part, now that particular the effect of gene can be nullified by there itself by making a gene function but will not express its negative effects onto the offspring. That is called gene silencing. Process, it is disabling the faulty genes, correct harmful mutation, insert a missing gene, or change activity of a normal gene. These are called gene editing. Normally, treating genetic disorders, gene editing is used. Sickle cell anemia, cancer, and HIV resistance, and muscular atrophy. These are the diseases generally with the help of gene editing will be done. Madam, I think another class is there, madam.
Yes, sir. Yes, sir. Shall we join again, sir? You can conclude it in 5
I
minutes.
think in another probably 1 minute I will conclude it.
Okay, sir.
Yeah. So this is embryo gene correction to prevent inheritance. This is gene editing, and those CRISPR Cas9 will be used. It is faster, cheaper, precise than going into other processes. In therapies generally it is used. Organ transplantation it is used. This is all done in vivo. That means you will not taking out anything. When the human body inside itself you are making this particular technique so that that particular gene is stopped from inheritance or stopped from expressing itself. With this, this is another called zinc finger technique, ZPT. ZPT, you simply remember whenever that particular, since the term is altogether different, if it forms in the sentence, zinc finger technique, ZPT, you remember, this is used to insert the correct gene of DNA into the parent through intravenous method. Zinc finger technique is a method used to insert the correct gene of DNA into the parent through intravenous technique. That means this is done in vivo. I told you in vitro. In vitro is taking out the uterus, doing changes, and then placing the embryo into the uterus. In vivo is in living condition only. You are not taking out anything, but you are doing this technique while in the parent itself, while it is living. You are not taking out anything. In vivo, that means in live condition. In vitro means in the lab. We told you about a CRISPR Cas9.